right, so today we're going to start a brief unit on membrane structure and synthesis. That's a hard word to say. Membrane structure and what is the plasma membrane made of? Phospholipids. We learned about phospholipids when we first started learning about water. We learned about phospholipids when we talked about macromolecules. We learned about phospholipids when we talked about cells. And today and next time we're going to have 2 whole lectures on phospholipids and why they're so important to promoting life. So we have a 2-lecture series. One is on membrane structure and synthesis, and the next one is on transport across the membrane. Brief review from the last lecture. The plasma membrane, the big point of the plasma membrane, this thing composed of phospholipids and stuff, is to separate the inside of the cell from the outside of the cell. It lets you have an inside and an outside, an internal environment and an external environment. It's also good for this is not going to work out well for me. It's also good for selective uptake. It's good for defense. It's good for cellular communication. There are all sorts of functions of the plasma membrane. All of these functions happen because of the specific form of those phospholipids, because of the way phospholipids interact with embedded proteins. So going through one at a time, we take up some things but not others. It is called selective permeability. It will allow some molecules in but leave some out. It's like you guys with your love lives. You want to keep some people in. You want to keep some people out. Actually, you're better not with your own love life. You're better with your friends' love lives. You're just like, you want them to keep people out because you know what's best for them. And they still don't listen to you. The membrane's important for compartmentalization. What are these individual little compartments inside the cell called? Organelles. That's the big group, right? Each organelle has its own unique environment. The lysosome, for instance, is more acidic. It's got a lower pH than other areas. It's able to maintain that specific environment because it is compartmentalized. It's good for protein sorting. We've got the Golgi apparatus, the endoplasmic reticulum. All of these are made up of plasma membranes. They help surround and bind proteins and move them from one part in the cell to another. The cell membranes are really important for anchoring the cytoskeleton. You have some sections that are actually embedded in the cytoskeleton. Those parts that are embedded in the cytoskeleton can be described as hydrophobic. They're always oriented away from water. The part that's inside of the cytoplasm, in the cytosol, are going to be hydrophilic areas because that's an aqueous solution. So you can anchor the cytoskeleton in the membrane. You can receive signals. The only way to get something from the outside of the cell to the inside of the cell, like a signal, is through the plasma membrane. So here we've got a protein on the outside of the embedded in the membrane. A signal comes in, and that releases some set of signals inside the cell to do something. It's its interface with the rest of the world. So, that's basically the membrane. That's what we talked about last lecture. Now we're going to talk about the structure of the membrane. Form determines function. Ultimately, the form of the membrane determines the function of the membrane. The plasma membrane is known as a fluid mosaic. Have you guys ever heard the term mosaic before? What's a mosaic? Yeah, it's you take all sorts of little pieces, you put them together, and you get a mosaic. Here we've got a bunch of tiles of different colors. You put the tiles together in a certain way, and suddenly you have an image, a cohesive image. I don't know if you, I don't know why this pops into my mind, but if you squint hard enough at tiles on like a bathroom floor, you start to see patterns and images. I don't know if you guys do that, but I sure know that I look at that occasionally. I'm like, huh, yeah, it forms a little shape there. You take all of these different colors. You form patterns, you form shapes, you form new motifs. So when you have these different types of components, in this case, you've got blues and yellows and greens, and they come together and they form link. If you're to look at it in a biological sense, you've got different tiles. The tiles in the membrane are going to be your phospholipids. They're going to be carbohydrates attached. What process are you going to use to attach a carbohydrate to a lipid or a protein? Glycosylation. You might have proteins. Between proteins, lipids, and carbohydrates, you have all the tiles you need to build a plasma membrane. By far, the most abundant structure in the membrane is the phospholipid. That's another reason they call it the phospholipid bilayer. Phospholipids make up the sort of background, the core of that membrane. And if you remember the structure of phospholipids, switch over to the next slide. You've got a hydrophilic head and a hydrophobic tail. So these are all these little ball things are phospholipids. And we've got these hydrophobic tails. Here, you've got a large protein spanning the whole way through. What can you tell me about this solution out here? It's an aqueous solution. Up there, an aqueous solution. What do you know then about this part of the protein? It's a hydrophilic region. Here you have hydrophobic amino acids, and then up there, more hydrophilic ones. That allows these proteins to be rooted in the membrane. So we've seen this picture several times. It all comes down to the amphipathic nature of the phospholipid. It's amphipathic. What does it mean to be amphipathic? You have polar sides and nonpolar. You both love and hate water at the same time. The polar head is going to orient toward water, the nonpolar tail away from it. This leads to the formation of micelles. It also leads to the formation of bilayers automatically without any energy being expended. But it's not just phospholipids. There's also proteins. And I know these look like balloon animals. I thought about making some for you, and then I decided against it. So we've got these proteins that span all the way through. Look at all these coils, these helixes. Here you're seeing secondary structures, and they serve a purpose. When you think of a coil, it's wrapped in a circle, a spiral. What you have is a whole bunch of amino acids and then a hole in the center. It's like, have you guys ever played with a slinky before? Okay, did you ever get it to really walk downstairs? Okay, a few people have. I tend to find that the stairs are way too far set and it only goes like 3 steps down, and then you're like, oh man, now I've got to start it again. In any case, if you've looked at a slinky, you can look through a slinky like that. Just hold it up to your eye and look through it. There's a hole in the middle, a tube. That's what these are forming, tubes. And then things can go through those tubes. You can drop something through the center of a slinky. So these proteins are embedded in the membrane. And those proteins can do all sorts of different things. These proteins are ultimately what give the protein its different functions. Here you've got a hydrophobic region and then a hydrophilic and hydrophilic up there. If something binds here, it can cause a reaction on the other side. You can have communication back and forth with the exterior and interior of the cell. Attaching to these proteins and these lipids are going to be carbohydrates. And again, that's through the process of glycosylation. So glycosylation happens. We know that. Don't really know why it happens, but it definitely happens. It's great. If you read through the book sort of glosses over it. They're like, and there's glycosylation. Doesn't tell you why. There are a lot of hypotheses on why. Some decent explanations. I'll explain some to you today. But none of them are for sure. I have not found literature to back up a generally accepted reason for glycosylation. So when you add a carbohydrate to a lipid, you form a glycolipid. When you add a carbohydrate to a protein, what do you think you form? A glycoprotein. Because what does glyco mean? Sugar, carbohydrate. So through the process of glycosylation, you can make either glycolipids or glycoproteins, which can interact in the membrane. All right, so now we've got our pieces. We've got carbohydrates, lipids, and proteins all interacting together. These membranes are constantly moving. They're constantly shifting around. Imagine, if you will, a bunch of, tennis balls on a table. And the table starts to shake. The balls move. They roll past each other. They don't bounce all over, but they can shift around each other. That's what the phospholipid bilayer is like. It's constantly moving in these two dimensions, left and right, forward and backwards. But there are two sides to it. So while you have one table shaking and tennis balls moving all over it, the same thing is happening on the other side. So both sides are sort of shifting all the time, moving like a sea. If you think of, putting a few boats on water. As the boats sit on the water, they'll drift around, moving back and forth next to each other. They don't go under each other. They always shift next to each other. You can actually flip, though. You can get from one side to the other. That's why I have this note at the bottom. Tell them about flipase. There's this enzyme called flipase. Flipase helps things flip. In order to move a phospholipid from one side to the other, it's very difficult. Why would it be difficult to get a phospholipid from up there to down here? It's amphipathic, and you have to get through this hydrophobic layer. It's hard to do. It becomes much easier if the flipase comes and sort of puts a bag over that hydrophilic head. Now it can easily slide through, get to the other side. But for the most part, most of this movement is lateral, not up and down. So we've got these constantly moving phospholipid membranes. It's really easy to think of the cell as something that's static, that doesn't move, but it's constantly moving. Every part of it's constantly in motion. Within the membrane, you have different types of proteins. You have integral membrane proteins and peripheral membrane proteins. Integral membrane proteins are integrated into the membrane. Integral are in the membrane. Here, you've got the center one is an integral transmembrane protein. It goes all the way across. And it's integral because it's integrated into that phospholipid bilayer. There's a hydrophobic section of it. This one on the far right is still an integral membrane protein. It's not trans. It doesn't go all the way across. But it is rooted in the membrane. These integral membrane proteins are going to be important for cell signaling. They're important for cell transport, getting things in and out of the cell. The other kind of protein are peripheral membrane proteins. These are proteins that are not actually integrated into the membrane, but associated with it. This protein down here is connected to an integral one, but it itself is not integral. The one up at the top, in the center, that's a peripheral membrane protein. It is associated with the membrane, but it's not actually in the membrane. Most of the time when we're talking about membrane-bound proteins, we're talking about these integral proteins. But we can't forget that there are peripheral membrane proteins that do things as well. They might serve as a glue, they might serve as a messenger, they might do all sorts of different, they might have all sorts of different functions. So between these lipids and these proteins, you can have areas of the membrane that have specialized tasks. You might have doors into the cell or windows into the cell. You might have regions that are just really good for hooking onto other cells. Everything is not everywhere on a cell. Everything has its proper spot, even on the membrane. So what these protein complexes will do is start to form groups. And these groups stick together. Can I use your Dr. Pepper? Thanks. And your drink. Oh, it's such a cute little thing of water now. So you've got these two things are going to need to be associated with each other. And my shoe. All right. We've got three things that work together. Normally, they would just be floating all over the place, next to each other, sliding away. But when these things work together, they're going to become associated with each other. They're going to stick together. They adhere to each other. They form one unit so that they can work together evenly and work together well. One thing that can connect them are carbohydrate strings. If you have carbohydrates lashing these proteins together, you can form one cohesive factory unit. Okay, so do you guys remember a show called Gilligan's Island? Okay, three of you. Hey, raise your hand. Gilligan's Island. Oh, thank God. Okay, you know what I'm talking about then. Gilligan's Island. These are, for those of you who don't know, these people were stranded on an island due to absolute incompetence on the part of the first officer, the first mate, and ended up being stuck there for a long, long time. Now, on this island, there's no, what was it? No boats, no lights, no motor cars, not a single luxury. They had to get off. To get off, they needed to make a raft. Every single episode, they had to make a raft. They had bamboo. How are they going to hold the on Gilligan's Island? How do they hold the bamboo shoots together to form a raft? If you were stranded on an island and you needed to make a raft, important life lesson here, guys. You use vines, right? Yeah, that's right, because all you had were sticks to play with. You know this. I know. Engineering feats. So you've got these sticks that are held together with vines, and you build a raft. If you didn't have the vines, what would happen when you went to step on the sticks? They separate, and you've done that before, haven't you? You had you were at a lake or something, you put something, you're like, I'm going to step on this, and everything falls apart. And if you haven't, again, you just haven't lived. Those vines would be the carbohydrates that connect the proteins together. The proteins themselves are separate. You can hold them together using some sort of adhesive like carbohydrates. Now, how are you going to get carbohydrates onto these lipids and these proteins? Glycosylation. So there's one possible purpose for glycosylation, is to form the glue that holds these protein rafts together. And then the protein rafts sort of hang out together, floating along that phospholipid sea. A plasma membrane is typically termed a semifluid, not a fluid. In a fluid, you can move in three dimensions: up, down, left, right, forwards, and backwards. In a semifluid, you can only move in two directions: forward, backward, left, and right. It's sort of the difference between thinking of a submarine. A submarine can go underwater, above water, to the left, the right, forward, and backwards. A boat, on the other hand, really shouldn't be going down. Does that make sense? Anything that a boat can float on would be like a semifluid. If you're thinking of a submarine or an airplane, that would be more like a fluid. So what determines the rate of the fluidity of this ocean, of the semifluid? And it all comes down to the phospholipids. Length of the tails in phospholipids matters a lot. The longer the tail of the phospholipid, the more fluid you are. If you have two phospholipids, and they both have relatively long tails, these are going to have more fluidity than something that has a much shorter tail. Here. Actually, hold out your hand a second. So if I'm holding her hand, we got long, long, long tails, I can do a whole lot of movement. But if I have shorter tails, I can't do as much. Does that make sense? The longer the tail, the more fluidity you have. Kinks, the kinkier it is, the more fluid. All right, so by kinks, remember these phospholipids have a double bond in one of their tails. So if they have a small kink, these things are sort of right next to each other. They're not moving around much. It's sort of like when you're squished into an elevator and shoulder to shoulder. You can't move much. But if the kink is located farther up, suddenly the two phospholipids have to be much farther apart. Again, think of yourself in an elevator, but instead of you having your arms down, now your arms are out like this, and you're sort of swinging around. Nobody's going to get near you. You have a lot more motion, a lot more fluidity. You can do more things. You can move around more. So the kinkier it is, the bigger the kink in the tail, the more fluidity you have. And finally, the amount of cholesterol present has an impact. Cholesterol stabilizes your membranes. Now, at this point in your lives, you've probably heard how terrible cholesterol is for you, right? You've heard that before. Okay, just making sure. Yes, excess cholesterol is bad for you, but you do need a certain amount. Without cholesterol, at high temperatures, your cells would fly apart. They stabilize at low temperatures, cholesterol acts as a lubricant. Cholesterol acts like a grease, allowing the phospholipids to slide past each other. So 3 things impacting the fluidity of the membrane. How fast that membrane's moving around are going to be the length of lipids, the location of the double bond allowing for a kink, and how much cholesterol is present. So we've got movement. Sometimes proteins are floating along that semifluid. That's what we've been talking about till now. They're like little rafts floating along, and then wherever they happen to be, they do their function. Other times, they're anchored in one spot relative to the inside of the cell. There might be an organelle right underneath them, and they need to stay above that organelle. How would they stay tethered to the organelle? How can this protein on the outside of the cell stay in a relative position to something on the inside of the cell? Any thoughts? So the cytoskeleton is going to be responsible for keeping those proteins anchored, keeping them in one spot. What component of the cytoskeleton gives the cell its form? There's a piece of the cytoskeleton that gives it its form. The cell, its form. Yeah. Okay, there are 3 parts of the cytoskeleton. Let's go with that. 3 parts of the cytoskeleton. What are the 3 components of the cytoskeleton? Microtubules, intermediate filaments, and actin filaments. Which one of those is all about structure? Intermediate filaments. So you have these intermediate filaments that these proteins might be anchored to. Because remember, the intermediate filaments are bound into the membrane, into this, into the hydrophobic region. The protein will also be bound into the hydrophobic region. So by attaching the protein to a filament to an intermediate filament, now it's not going to move relative to whatever else that filament's attached to. So some proteins float along the membrane, other proteins are anchored. So that is the structure of the membrane. Form determines function. We've got these phospholipids that are amphipathic, interacting with proteins, both of which may have carbohydrates. They're constantly shifting around. allowing some things into the cell and not others. Next part of today's lecture, how to build the membrane, membrane synthesis. So we need to build a phospholipid. And you have to build a phospholipid in 7 steps. First off, where do you build a phospholipid? What organelle is responsible for building phospholipids? There's an organelle. It builds things. It is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is where you're going to build phospholipids. So all of this happens there. First step, get all the pieces together. You can get pieces from you can take pieces in from the outside. Your lysosome's recycling things. You might break down other cells. You might break down other membranes and recover the monomers. Or you might build them from scratch. You might bring it in from outside the cell. You may recycle your own. All that matters at this point is you need to get all of those parts together. This is to build a phospholipid, which goes on to build a membrane. Next step is to meet at the endoplasmic reticulum. All of the parts needed for lipid synthesis have to be moved to the endoplasmic reticulum. How are you going to move these parts around a cell? Vesicles. So a vesicle can form around these pieces, creating a little transport module. That vesicle will move along what component of the cytoskeleton? The microtubules. But it's not going to move all by itself on the microtubules. It needs something else to move it from point A to point B. It'll use motor proteins to do that. So you're moving these vesicles filled with these parts to the smooth endoplasmic reticulum, where you're going to build it. So I have this picture. Right. The reason I have this picture up here. Sorry, I was like, why do I have that picture there? This is a great picture about 2 people meeting, running into each other. Do you guys know what this is from? I think it's a Pulitzer Prize winning picture. It was after World War II, I want to say. A sailor came back. The story is the sailor came back. He found his girl, and he kissed her. And that was all over all the papers, right? But that's not really what happened. And you can tell a lot just from this picture. Look at the body language. What does her arm tell you about what she thinks of this? She's about slapped in the face. She's not comfortable. Yeah, this guy really just came up from her behind, tipped her over, and kissed her. So what was an iconic image of somebody coming back, 2 people meeting, and typically people associate this with happy, lovely feelings, is actually evidence of assault. So now you know. There's the story. Turns out afterwards, this is recently in the past, 10 years, she said, no, I'm okay with it, don't worry about it. But still, tells a lot. There's always more to an image than just the story you hear. So meeting up at the endoplasmic reticulum. From there, you end up with an enzyme called coenzyme A. It's going to attach to the fatty acid chains. Remember, phospholipids have 2 fatty acid tails. You need to get this coenzyme A to attach to them, and that turns them on. It activates them. So here's a cool secret I'm going to tell you. In 1, 2, 3, 4, 5 lectures, I'm going to turn to you guys and say, hey, we're learning about coenzyme A today. Where else in the course have we talked about coenzyme A? And then you guys are going to stare at me blankly for a few minutes, and I'm going to say, I better stop. I'm going to go get a drink of water real quick. You guys find it, and then you'll flip through your notes while I'm out getting a drink of water, and I'll come back, and then somebody in the first 2 rows will answer, 3 rows, will answer where we've seen coenzyme A before. The answer is right here. Coenzyme A is helpful in building phospholipids. So you've got these 2 fatty acid tails. you're going to stick them onto a phosphate molecule, a phosphate glycerin molecule. Phosphate, where have we heard that before? We keep seeing phosphates again and again. Where do you guys remember phosphate from? From where? The carbon lecture, yeah. Where in the carbon lecture? That's right, the organic molecule lecture. We talked about them at the very beginning. We talked about carbon has these 4 properties. Carbon can have many different forms. And one more thing, carbon binds to what? Functional groups. So phosphate is one of their functional groups. Here it's binding to carbon, just like we said it would. And that forms a little head. So now that you've got these fatty acid tails that are hydrophobic and this polar head, it's going to stick itself into the membrane. These tails want to get away from water. So it sort of inserts itself into this hydrophobic region with that phosphate sticking out the top. Then another molecule comes along, an enzyme called phosphatase comes and hacks off the phosphate, slices it off. And that's kind of a good name for an enzyme, phosphatase. When you think A's, think enzyme. And what does it do, this phosphatase? It removes phosphate, yes! When in doubt, if it says A's at the end and another word in the beginning, it usually means it removes whatever that thing is in the beginning. Then once you cut off it, you come back in and stick a choline molecule on there. It's charged. And that attaches to the phosphate, with a different phosphate group attached, sticks on the glycerol. So now you have a polar head and non-polar tails creating what kind of molecule? Amphipathic. And that's where you get your phospholipid. Any questions? Here's the other cool thing. I'm not going to ask you anything about that on the exam. But if I told you that at the beginning, none of you would have listened to what I was saying, and I think it's really cool. What will be on the exam, though, is how to build a transmembrane protein. I can almost guarantee that's going to be a straight-up question, like one of those 3-point questions. You need to build a transmembrane protein. Transmembrane proteins are, again, the ones that go all the way through the membrane. You can do it in a few different ways, but the most predominant one is using the endoplasmic reticulum. Remember, one of the properties of the endoplasmic reticulum is to embed proteins in the phospholipid bilayer, building a protein that goes all the way through. On its surface, it sounds kind of difficult to do. You have this thing that's hydrophilic, hydrophobic, and hydrophilic. How do you get that hydrophilic part through the hydrophobic center? You don't have to. You form a channel. So what ends up happening is, in the rough endoplasmic reticulum, you have a C-shaped protein, sort of just hanging out in the endoplasmic reticulum. Another C-shaped protein in the, this is the puppet show, by the way. Another C-shaped proteins in the endoplasmic reticulum, and they come together like that to form a circle, a hole. So you have one here floating around, one here floating around. They meet up, and now you've got a pathway through the endoplasmic reticulum. What can bind to that is a ribosome. The ribosome has just come from the nuclear pore. It binds to that pore, that hole in the endoplasmic reticulum. After it's bound, back to taking my shoe off again. All right, so here's the ribosome binds to the endoplasmic reticulum, which apparently now is this table. The RNA is going to come along and bind to the ribosome. Let me use your notebook. It's not like you're taking notes. There we go. All right, so you've got lots of information here, right? That binds to the ribosome. Bind. Stay bound. Good. So that binds to the ribosome. This gives you all the instructions you need to build a polypeptide. So far, so good? The polypeptide is going to be produced from that ribosome and fed through the hole into the lumen of the endoplasmic reticulum. So you begin to thread this long polypeptide down in here. All right. Peptides all have different charges to them. You have some that are charged, some that are uncharged. The ones that are charged, that have a positive side or a negative side, are going to be polar. How will they interact with water? They'll dissolve in it. They're hydrophilic. They love it. So if you have polar amino acids, they're going to come out here and love this aqueous solution down here. If you have nonpolar ones, hydrophobic ones, they're going to get to the bottom of that hole, that channel, the pore, and they're going to hit that aqueous solution and stop because they don't want to keep going in. They are scared of water. They're like, no, to hell with this. I'm not going to go into the ER. So they just stay in the pore. When you get a chain of about 20 of those that refuse to go farther down, suddenly there's too much pressure that's built up in the under the ribosome, and it pops up. And I'll totally destroy everything here. So here's your RNA. There you go. The ribosome then pops up, trailing behind it a whole bunch of hydrophilic amino acids. Once you've got that, now you have hydrophilic amino acids here, hydrophobic ones here, hydrophilic ones here. You get to the end of the chain, and the ribosome pops off. Okay, think of it a different way. Have any of you guys ever accidentally flooded a toilet? Okay, good. Just making sure. And if you haven't, you're probably lying, and you did it at a friend's house, and you panicked. That's the worst, isn't it? You go to a friend's house, and you accidentally clog their toilet, and you're like, oh, what am I going to do? And then you're like, quick, I need a plunger. Well, they don't have one conveniently around. Just watching this toilet fill up, and you have to blame somebody else. Like, you should see what somebody else did in there. Disgusting. Not that I've done that. So if you've flooded the toilet, you understand the basic property behind this. Starts off, some of the things start going down the drain. Those would be like those hydrophilic ones at the beginning. They're going downward. Eventually, you're going to have too much clogging up that hole so that nothing else can get down. So instead, which direction does the water go? Yeah, onto the floor. It goes up and out. Just like our amino acid sequence. So we've got amino acids down here that are hydrophilic. You clog up the hole with your hydrophobic ones. Pressure builds up, pops the ribosome off, trailing behind it a bunch more hydrophilic amino acids until you have a full transmembrane protein going all the way through the endoplasmic reticulum. And then that can do all sorts of cool things. It can act as a signal. It can act for adhesion. It can act for membrane transport. But it works. Any questions? Yes? The protein is now embedded. Now it's stuck. You can't get it out because you got this hydrophilic region. So this part loves water. You can't pull it back out because that hydrophilic region really wants to stay down here. The hydrophobic region is stuck in the hydrophobic center of the phospholipid bilayer. So it's not going to move. It can't go down. It's going to hit water. It can't go up. It's going to hit water. And then the other hydrophilic region at the top can't be pulled through the hydrophobic layer because it's hydrophilic. So it is completely embedded. It's not going to move. It's like cemented in place now. It can move back and forth within the phospholipid bilayer, but it can't escape the phospholipid bilayer. Yes? This is exactly what that is. This is explaining the process of how the rough ER takes the ribosome and pushes the polypeptide in. It is, because the ER is made of plasma membranes. Yeah, pretty nice, huh? All those organelles are made up of this. It's all coming together. Questions? There you go. So that's the plasma membrane. So there we go. That is the short lecture about the membrane. The form of the membrane, everything we talked about today is what gives rise to the functions of the membrane. Next time, we're going to talk about how things get in and out of the cell, what gets in and out of the cell, and when, and in what quantities. I would suggest reviewing moles and molarity, because we're going to touch on that in the next lecture. And we're also going to talk about osmosis, diffusion, and active transport. If you could have some pre-knowledge coming in, that will be most beneficial to you because you may have some questions on how this works. In the next lecture, we're going to have a lot more practical application work rather than just memorize how things are built. The chemistry behind these phospholipids gives rise to the traits of the plasma membrane.